Have you ever noticed the warnings in drug commercials about fungal infections? They often mention rare but serious infections. These warnings are not just filler, they're crucial. Dimorphic fungi like Histoplasma and Blastomyces are behind some of these infections. They might sound exotic, but they're present in our environment. Such fungi can cause significant health issues if not diagnosed correctly. In medical settings, understanding these fungi is essential. They can mimic other diseases, leading to misdiagnosis. Dimorphic fungi change form depending on their environment. This trait makes them particularly tricky to identify. Histoplasma and Blastomyces exhibit distinct mold and yeast forms. In their mold form at room temperature, they produce hyphae and conidiophores, with conidia that vary in shape. When in yeast form, they transform into yeast cells with unique morphologies. Histoplasma yeast cells are small and oval with a pseudocapsule, while Blastomyces yeast cells are larger and have thick walls with broad-based buds. Awareness of these fungi and their forms can help in early diagnosis and treatment. Mycologists, medical students, and pathologists must remain vigilant. They need to be well-versed in the characteristics and behaviors of these fungi. This knowledge is crucial for effective diagnosis and management of infections they cause. Additionally, understanding doppelganger organisms that mimic these fungi is essential for accurate diagnosis. Histoplasma capsulatum is a dimorphic fungus. This means it exists in two forms, mold and yeast. At room temperature, it grows as a mold. The mold form is characterized by filamentous growth, known as hyphae. The hyphae are delicate and produce conidiophores, which bear round tuberculate or knobby macroconidia. It also bears microconidia, which are typically small, oval, and smooth-walled. When the temperature is raised to 37 degrees Celsius, it transforms into yeast. The yeast form is unicellular, small, and possesses a pseudocapsule. In reality, the pseudocapsule is nearly irrelevant as it is often impossible to see. It's important to note that histoplasma has doppelganger organisms, which can look similar under a microscope. The most common is Cepidonium species, which also possesses the tuberculate macroconidia. Recognizing these similarities is crucial for accurate diagnosis and treatment. A thorough understanding of the fungal life cycle and its environmental sources is critical. It equips medical professionals to better anticipate and identify potential cases of histoplasmosis. Diagnosing histoplasmosis presents a unique set of challenges. The symptoms often resemble other respiratory diseases. This overlap complicates the clinical picture. Moreover, traditional diagnostic methods can be inadequate. Histoplasma's ability to mimic other fungi adds to the complexity. Identifying the mold forms and the conidiophores can be challenging enough alone. Then, the need to confirm microscopic findings with fancier methods, such as MALDI or DNA sequencing, can add additional delay and cost. Serological tests can also cross-react with the dimorph Emergomyces, leading to false positives. Cultures, although definitive, are time-consuming. Histopathology requires expert interpretation to identify the yeast forms amidst surrounding tissue, which can also resemble Emergomyces. Molecular techniques are emerging as valuable tools. Polymerase chain reaction (PCR) assays offer more rapid and accurate results. These tests can specifically identify histoplasma DNA. However, the availability and cost of these tests pose challenges. Not all medical facilities have access to them. Despite these advances, diagnosing histoplasmosis often requires a combination of methods. Each tool provides a piece of the puzzle. The role of pathologists and mycologists is crucial here. Their expertise in interpreting results can guide the diagnostic process, ensuring accurate identification of the infection. In recent years, a new player has entered the scene, Emergomyces. This emerging pathogen is gaining attention in the medical community. Like histoplasma, it is a dimorphic fungus. It poses a significant threat, especially to immunocompromised individuals. In its mold form, Emergomyces produces hyphae and conidiophores that resemble other fungi. The conidia are typically small and oval, with some in whorls. When it transitions to its yeast form, the cells are small and often resemble histoplasma. Clinically, Emergomyces infections resemble other fungal diseases. This includes symptoms like fever, cough, and weight loss. In severe cases, skin lesions and organ involvement can occur. These overlapping symptoms complicate diagnosis further. The emergence of Emergomyces underscores the need for continued vigilance. Mycologists and pathologists must stay updated on its characteristics. Understanding this new pathogen is vital for its early detection and treatment. It's also important to distinguish Emergomyces from doppelganger organisms that present similar features. This knowledge can prevent serious health outcomes in affected individuals. Section 5. Unveiling Blastomyces, dermatitidis. Blastomyces dermatitidis is another significant dimorphic fungus that calls the Ohio-Mississippi River Valley home. It shares similarities with Histoplasma but also has distinct features. Understanding its morphology is crucial for accurate diagnosis and treatment. At room temperature, Blastomyces dermatitidis grows as a mold. The mold form features delicate hyphae and lollipop-shaped conidiophores, with small, round to oval conidia. This form may be mistaken for an immature histoplasma mold. When the temperature rises to 37 degrees Celsius, it transitions to its yeast form. The yeast form is characterized by broad-based budding, which is a key diagnostic feature. This dimorphic nature allows it to adapt and thrive in different environments. Accurate identification of these forms is essential for proper treatment. Like histoplasma, the colonies of Blastomyces are small, slow-growing and glabrous to spiky. Ensuring correct identification helps in administering the right care, reducing the risk of complications. It's also important to distinguish Blastomyces from doppelganger organisms that may appear similar under the microscope, such as Chrysosporium and Pseudosporium. Section 6. Clinical picture of Blastomycosis. Blastomycosis presents a unique clinical challenge. The mold form of Blastomyces dermatitidis grows as hyphae, which produce conidiophores and conidia. Conidia are small, round to oval spores that can be inhaled, leading to infection. Once inhaled, the conidia transform into the yeast form in the lungs. The yeast cells are large, thick-walled, and have a characteristic broad-based budding. These yeast cells can cause symptoms that mimic other respiratory infections. Common initial symptoms include fever, cough, and fatigue. These are often mistaken for bacterial pneumonia or tuberculosis. As the infection progresses, more severe symptoms can develop, including weight loss, night sweats, and chest pain. In some cases, the infection spreads beyond the lungs. Disseminated blastomycosis can affect the skin, bones, and central nervous system. Diagnosing blastomycosis involves several techniques. Cultures and microscopic examination are standard, but often need to be confirmed with molecular methods, MALDI, DNA probes, or yeast-to-mold conversion. Serological tests have limited use due to cross-reactivity with other fungi. Molecular methods, such as PCR, are promising. They offer faster and more specific detection. However, like histoplasmosis, these tests are not yet universally available. This highlights the need for ongoing research and development in fungal diagnostics. It's also important to consider doppelganger organisms that can mimic Blastomyces in clinical presentations. These include other dimorphic fungi like Histoplasma and coccidioids, which require careful differentiation. Section 7. Mycologists, the Unsung Heroes. Mycologists play a vital role in diagnosing fungal infections. Their expertise is crucial in identifying dimorphic fungi, including their mold forms. They study the hyphae and conidiophores, which are essential for fungal reproduction. The shape and structure of conidia provide critical insights into fungal identification. In the lab, mycologists also focus on the yeast form of these fungi. They analyze yeast cell morphology to differentiate between species. This detailed work ensures accurate diagnosis and treatment. Mycologists encounter a variety of rare and doppelganger organisms that mimic pathogenic fungi. Beyond diagnosis, mycologists contribute to public health. They study the environmental sources and spread of fungi. This research informs prevention strategies and public awareness campaigns. By understanding fungal ecology, they help reduce infection risks. The role of mycologists is often overlooked. However, their work impacts both individual patient care and broader public health. As the field of mycology evolves, their contributions will continue to be invaluable. Section 8. Navigating the mycological landscape. The landscape of mycology is ever-changing. Understanding the mold forms, hyphae, and conidiophores is crucial. Histoplasma and Blastomyces exhibit unique conidia shapes. Experts must adapt to these developments to stay ahead. Technological advancements offer new diagnostic possibilities. Molecular techniques are becoming more accessible. These tools provide quicker, more precise results. They are transforming the way fungal infections are diagnosed. Education and training are crucial in this evolving field. Medical students and pathologists need to be well-versed in mycology. Continuous learning ensures they are prepared for the challenges posed by dimorphic fungi. Professional development opportunities must be accessible and engaging. Collaboration is key to navigating these changes. Mycologists, pathologists, and clinicians must work together. By sharing knowledge and resources, they can improve patient outcomes. This teamwork is essential for advancing the field of fungal diagnostics. Section 9. Conclusion. The future of fungal diagnostics. The future of fungal diagnostics is promising yet challenging. Dimorphic fungi like Histoplasma and Blastomyces will continue to pose threats. However, advancements in technology and collaboration offer hope. These tools and partnerships will enhance diagnostic accuracy and treatment outcomes. Mycologists will remain at the forefront of this field. Their expertise is indispensable in understanding and managing fungal infections. They will continue to play a crucial role in research, diagnosis, and public health initiatives. As new pathogens emerge, the importance of mycological knowledge grows. Staying informed and adaptable is key for healthcare professionals. By embracing new technologies and collaborative approaches, they can effectively combat fungal infections. In conclusion, the world of dimorphic fungi is complex and dynamic. With the expertise of mycologists and advancements in diagnostics, the future looks bright. Stay curious and continue to explore the mycology niche, or as I like to call it, the Chitin Corner.